Hello, this is an introduction to primer design from the iGEM team at Duke University. Okay, so principally primer design is for PCR, and primers are just short strings of nucleotides that bind to the larger DNA that you want to amplify or modify in some way, depending on your specific purposes. These can range from mutagenesis to simply amplification. And it occurs through a polymerase chain reaction, which is something that fundamentally follows the same pattern over and over again, and just occurs based on what you've put in there, which is DNA polymerase, nucleotides, and the primers. This allows you to consistently amplify sequences over and over again so that you eventually have far more DNA than you started out with. Something on 2 to the nth times, where n represents the number of cycles you've put the PCR reaction through. And what you will want to do is, depending on your purposes, you'll select a region of DNA that you'd like to use as your primer. There are certain rules that you have to abide by. These can be found on the Primer Design Guide from Premier Biosoft. These rules are somewhat lax, and that means you have a little room to play with them. There's a little room with the primer annealing temperature, which determines what temperature the primers will anneal to the DNA at and allow you to amplify certain regions of the sequence that you want. The GC content is going to play a role in that. Higher the GC content, the higher the melting temperature, and you want the melting temperature to be around somewhere, well, the annealing temperature is going to be somewhat higher. So you want the temperature to be at around 64 degrees. You can play with that a little bit as you will, but it really depends on what you have, what you need, and what you're willing to go through. There's also secondary structures that can occur in the primers, which will make amplification and just the DNA polymerase being able to amplify anything that you want very difficult. In order to do that, go to the IDT-DNA.com analyzer and plug in your sequences. These can look at hairpin, self-dimers, heterodimers, and melting temperature mismatches. Elongation occurs at 72 degrees Celsius, so you really want your melting temperature to be kind of under that, but again, there's room to play with. The mix of this reaction, as I said, contains nucleotides and DNA polymerase as the most important components. And because DNA polymerase can only operate in a single direction, there is a certain degree of control that we have over how and what gets amplified through the creation of our primers. Certain things that you got to be aware for are repeats and other stuff, and those can be found at the previously indicated resources here and here. When you go check melting temperature, you're going to use the NEBTM calculator, which will give you the melting temperature for each primer and their annealing temperature. Now that we've covered the basics, I want to go into and talk about how you design primers, which is relatively simple. I use Seq Builder from DNASTAR, and what we have here is an example of a DNA sequence that you can create primers for. And as we will be discussing here, we have a top strand primer, and primers are going to be given by a directional arrow. As you can see here, this top strand primer points in one direction, and if we look here, we have a bottom strand primer. Both of these will bind to different segments of DNA, and because Seq Builder has the function which gives you the arrows, you can see which direction the DNA polymerase will build in. And based on the structure of these two primer designs, you're only amplifying regions from the start of the primer over here to the end of your bottom strand primer over here. It allows you to amplify a specific region of DNA and essentially get more. It's relatively simple, and that's what PCR reactions were originally intended for. However, people have since modified the PCR reactions such that different things can occur. For instance, if you are working with a plasmid and you create a bottom strand primer here and create a bottom strand primer, or a top strand primer rather, over here, you'll notice the arrows point in opposite directions. That means you amplify the whole sequence. And with mutagenesis and insertion, you can actually add an additional DNA sequence in between those two primers to allow for insertion of that DNA sequence within the larger schema of what you're working with. That makes modification of DNA rather simple in some respects. You can also forego that, and depending on what you have and what you do, you can actually delete sequences between those primers. So in this area right here, if your primers were spaced further apart, for instance, you could delete those entire regions of DNA and basically cut and paste. Essentially, PCR in this way will allow you to cut and paste certain things in and make completely different DNA, or rather modifications thereof of your existing DNA. So just going over what we've already covered, amplification primarily serves the purpose of making more DNA, and specifically certain sequences of that DNA. It allows you to amplify certain regions and look for things that you typically wouldn't be able to look for because you have either too many different areas, like too large sequences of DNA, or not enough DNA. It lets you amplify the entire DNA sequences if you have a circularized plasmid. With regard to mutagenesis, you have multiple methods to do this, and insertion, deletion, and other variants are also bound. Primer design is relatively simple as long as you keep in mind the resources that you have. It's relatively easy to design primers, and it never really works the first time. It'll take a few times of practice before you get it right, but it's a reaction that is carried out in just about every lab in the world for biology. And quite frankly, it's very useful. Thank you, and have a nice day.